Wait, did someone join? Oh, Jake Pessin. Oh, that's a researcher. Okay, I need to wear a blazer.
All right, good morning or good afternoon, everyone. Thank you so much for coming to our diabetes conference, US Diab Con. I'm Alex Yurkovich, president and co-founder of Seeking a Path Forward for Diabetes, a recognized student organization at University of Southern California. At SED, we strive to raise awareness about diabetes, promote the treatment of diabetes, and prevent the onset of diabetes through guest speaker seminars, going to local diabetes events, creating social media posts, giving out free over-the-counter diabetes medication, doing free A1C tests, and teaching others how to administer severe hypoglycemic first aid. Now, what we hope to do here at US Diab Con is to give US students and faculty the opportunity to understand what is happening in the world of diabetes in the US by giving a platform for US diabetic experts to discuss what they are working on or have worked on. Furthermore, this event will help connect university students and faculty throughout the US who are interested in diabetes with one another. Today, we have a wide range of speakers from all throughout the US, from 7 different educational institutions, discussing diabetes biology, diabetes population health, diabetes clinical care, and diabetes technology. Each speaker will give a presentation about what they are working on / have worked on for about 10 minutes, and then we'll answer your questions for about 5 minutes. Furthermore, during the middle of the event, there will be a networking segment for about 20 minutes where event attendees can network with the speakers. There will be 4 breakout rooms with 1 breakout room for each category, so 2 speakers per breakout room. An executive officer or member from our organization will be moderating the breakout room, and we'll display questions that the attendees can potentially ask the speakers and vice versa to facilitate conversation. Lastly, at the end of the event, there will be a very short fundraising segment to help raise funds for our organization, which will go to support our activities. You can scan this QR code for a full itinerary of the event with rough time estimations, which is also found in your RSVP email. I'll give you guys a few seconds to scan this QR code. Awesome. Our first 2 speakers will be speaking about diabetes biology, more specifically the fundamental biological mechanisms underlying diabetes and its related complications. Now, let's introduce our first speaker. Our first speaker will be discussing what he has worked on in the field of diabetes biology. He is an associate professor of medical sciences at the Naomi Berrie Diabetes Center at the Columbia Vagelos College of Physicians and Surgeons. He obtained his BS degree in biochemistry and MS degree in microbiology from the University of Nancy in France, and received his PhD in immunology from the University of London at University College London. He subsequently trained at Stanford University, where he was consecutively postdoctoral fellow, research associate, and instructor in the Department of Medicine, Division of Immunology and Rheumatology. He joined Columbia University in November 2012, and his research interests revolve around the pathogenesis and prevention of type 1 diabetes. He and his group study how several processes that contribute to the maintenance of immune tolerance are impaired, allowing the progression of the disease. The lab works on several new therapeutic strategies aimed at restoring immune tolerance and blocking autoimmunity. Today, he will be discussing antigen-specific tolerance in type 1 diabetes. Without further ado, it is an honor to welcome Dr. Remi Creusot.
Hi, everyone. Can you see my full screen? Can you hear me?
Yeah, we
Yes,
can
we
see your
can hear
screen and hear you. Yep.
And it's a full screen, right?
Correct. Yeah.
Thank you. Thank you for the invite. It's a very pleasure to get to talk to you guys about what's being done in this field I'm working on. It's going to be a pretty general presentation, and towards the end, I'll tell you a little bit about what we're doing in my lab. So just to start, I want to put on this disclosure, which is that I'm a scientific founder of a startup working in this area. So I want to start by saying that to explain what is antigen-specific tolerance. So T cells and B cells, they are meant to react against antigens, but when it comes to self-antigens, it's a very different it's a very different response because they need to have a completely different types of response, right? And so this is all in response to antigen presentation, but the way they see some antigens has to be in a completely different context. So let me give you an example, right? If you are an immune cells and the antigen is a rat, this is a context where you want to get rid of it. So this is basically what happens if the antigen is a viral antigen, for example. But in the case of a self-antigen, and is a rat, you should be able to tolerate it. And this is a context, a different context where the immune cells will be able to see these antigens are very perfectly innocent and actually very good to tolerate. So there are different contexts where the certain subtypes of cells are presenting antigen in an incorrect context. And that's what happened in autoimmune diseases and in type 1 diabetes in particular. So there are antigen presenting cells that engage T cells, for example, and provide and expose these antigens, these self-antigens like beta cell antigens, while responding to danger signals and inflammation. And these contexts are leading to the improper activation of these T cells, which will then turn against the tissue, like beta cells in this case. So antigen-specific tolerance, I would say there are three pillars. And one is the context, which is what I said earlier, type of cells that are presenting these antigen and the signals that they provide to the T cells will have to be adequate. There's the location, the tissue in which this happens, and environment is very important, and how long this presentation takes place. You know, when you have a viral infection, it's typically something that is short duration. But in the case of a self-antigen is always supposed to be there. So it's always supposed to be presented for the long duration. And this is despite of this being always there, there's a response against these self-antigens. So now more specifically in the context of type 1 diabetes, the question is, where are these antigens being seen by the immune system? And talking about beta cell antigens, right? So the first place is in the thymus, where the T cells develop. This is their chance to get to and to be exposed to these self-antigens. But the thymus only present a subset of beta cell antigens. And there, the context is pretty heterogenic. It's always, it's basically represented by the green background there, and the antigens are always presented. But this function decrease over time. And once the T cells are released from the thymus, they're not going there. So there's actually quite a few T cells that escape this sort of filter or the system that is there to purge potentially autoreactive T cells. And then where else are these antigens? Well, typically they are in the draining lymph nodes of the pancreas. And there, again, the antigens is always there, presented. But the problem is that over time, the context is changing from innocuous to pro-inflammatory or inflamed. And that's shown here by the change of color from green to red. And therefore here, the T cells that are now circulating in your body are always the only way they can be activated is in the context of seeing this antigen in the incorrect context. And I'm a bit oversimplifying there, but this is basically the idea. And then when it comes to try to rectify this issue, for example, antigen-specific immunotherapy, we are providing antigens elsewhere, potentially. So it depends on the mechanism. And these antigens will be presented in different tissues. And these tissues are supposed to present antigens in a correct context. And as you can see here, because we deliver these antigens, the source of antigen is the actual delivery, right? So over time, it goes away and you have to keep giving it back. And so that's what you see here. It goes up and down, and that all depends on how frequently you provide these antigens. And so what we think is that to achieve or to reestablish antigen-specific tolerance, we require persistent exposure, ideally at these other sites where there's no inflammation, of either key antigens or epitopes, which are fragments of these antigens, that is peptide presented to T cells. And potentially you may be requiring immunomodulatory molecule to help in terms of the signals that are given to these T cells. So when you have persistent presentation of antigen, ideally you want to go from a very highly intermittent exposure to a more continuous exposure of antigen. And we think that that way you have a more efficient induction of antigen-specific tolerance. And there are different ways to achieve that, either by eliminating the pathogenic T cells that are recognizing these antigens, or converting them into Tregs, or even in the context of B cells, if you're using 4 antigens, there's a possibility of inducing Bregs. So these are regulatory cells that have suppressive capabilities. So there are different tissues I mentioned earlier that are being targeted as sites for presenting those antigens. And in those tissues, there are a number of cell types that have been considered having a great potential in the ability to present these antigens in the proper context. Right? And to access these tissues or cell types, there are different ways of delivering antigens, either in the forms of proteins, of peptides, and even in the form of DNA or RNA. And here's just some examples of methods of delivery system, particularly including nanoparticles, for example, that can deliver all sorts of cargoes, protein, even nucleic acids. There are also cell therapies briefly mentioned at the end, like antigen-presenting cells that already have a capability of providing the correct signals, like tolerogenic dendritic cells, or even artificial antigen-presenting cells are just in the forms of beads or nanoparticles that have these MHC peptide complexes that T cells can recognize. And that seems to be sufficient to induce the effect that we want. So what are these outcomes that we are seeking? Obviously, when the self-antigens are being presented to T cells, one of the sort of effect is to induce a negative signals to turn off these T cells, right? A highly desired outcome is the conversion of CD4 T cells into what's called regulatory T cell, because these also have the ability to suppress other T cells in their vicinity. And that is basically what we call bystander suppression. Another important complex notion is the notion of infectious tolerance, which is this ability of these newly converted Tregs to go around and find another antigen presenting cells presenting the same antigen, but potentially presenting other antigens and interacting with additional T cells that are active to different antigens and also suppress them. So that's some sort of chain reactions where you can actually start to get suppression from a more limited set of antigens to a broader set of antigens. And overall, that we would characterize this as antigen-specific regulation. But in order to really get to antigen-specific tolerance, you would need this to persist long term. And basically, what we would call tolerance is when the T cells are properly trained, protecting such that, and it's trained on its own, and once you discontinue therapy, you would be able to still maintain the protection that you induced originally. So to get antigen-specific tolerance, you need to be able to discontinue therapy. All right, so now another important notion is that, and I think personally, that antigen-specific therapy for autoimmune diseases like this, where there's many different antigens that are targeted, is that it needs to be, to some extent, tailored to a patient or to a group of patients. One notion is that to get presentation, in fact, of the epitope, you need to have the right HLA haplotype, which is the type of MHC molecule. So a particular MHC can only bind a certain set of peptides. And so if you have critical key epitopes, you need to have the right MHC to present them. And so what we think is that the therapy has to be tailored to basically pick the right epitopes that such the person is able to present them. Otherwise, it's kind of useless. And even when you think about full antigens, where it can give rise to a large number of epitopes, there's evidence that certain antigens you have a better response to with people with particular HLA and not a good response with people of different HLA. So this is a characteristic of the patient that's very important to take into account. And so there are remaining challenges in this field, which is to what antigens are most appropriate for a group of patients with particular HLA. And also there are other patient characteristics that we call endotypes that it's important to take also into account in order to, when you think about designing these kinds of approaches for delivering the antigens and also defining what kind of antigens will be needed. So another important thing is you have to remember that when you start inducing tolerance or inducing regulation, it takes time to be established. And even if you reach a time when you can actually discontinue treatment and the patient will have maintained that tolerance long term, the disease in the meantime is still progressing. So there are maybe ways to accelerate this by enhancing the signals that are provided to the T cells through immunomodulation. So these are actually other things that you can provide on the side along with the antigen, or something you can encode if you use nucleic acids to improve the response or accelerate the type of outcome that you want to achieve. The other thing is you might want to think about posing the autoimmune reactivity against self-antigens, for example, with therapy like recently approved teplizumab. And so that if you have a way to pose the immune response, then you can allow antigen-specific tolerance to build up. And so one of the challenges is to find a way to achieve also persistent presentation of antigens, so you don't have to give it all the time or frequently to the patients, as opposed to a regular vaccine, which you give a few times. We think that these types of treatment, you would have to give it much more frequently. So a little bit about the landscape of the companies and technologies that have worked on trying to induce antigen-specific tolerance, not just in type 1 diabetes, but in a variety of autoimmune diseases. This is from a review from 2020. And looking at it right now, 5 years later, I can show you that already a lot of these companies have folded. And so it's not something that is very trivial. When you think about it in theory, it seems a pretty simple concept, but it's just not easy to achieve. But There are new companies that have emerged since then, and all of them have preclinical program in type 1 diabetes. And there are some of these other companies that have been there around for a long time that still are looking at type 1 diabetes, still at the preclinical stage. Some of them have reached the IND enabling phase, and one of them, in diamide, is already in phase 3 clinical trial. But those that are still in the earlier phase, fortunately, they have other indications that are in clinical trials. And so the hope is that if these clinical trials are successful, they will also move on to test it in type 1 diabetes patients. A few words about the stages of type 1 diabetes and because, you know, when is best to intervene and how. So this is my personal opinion. When it comes to stage 1, which is already hard to define, but basically patients who have a genetic risk but don't have sign of autoimmune disease, we want to, there's probably a lot of patients, but we want to be able to intervene very gently, thinking about potentially using oral tolerance and combining corrections input in microbiome. So to prevent diabetes, but in a very gentle way, basically. At stage 2, which is where there is very clear signs of autoimmunity, and also we can sort of estimate how far they are from onset of diabetes. I think this is a perfect time for employing antigen-specific therapies, and with or without the use of biologics to delay the onset of disease while reestablishing antigen-specific tolerance is pretty safe from the studies that have been done so far. Once you have been diagnosed with diabetes and you have limited residual beta cell mass, it's probably more difficult. So you might want to accelerate the induction of antigen-specific tolerance with more aggressive types of therapies, and really trying hard to protect those few residual beta cell mass that you have, but it's very difficult. And finally, in long-standing type 1 diabetes, where we probably mostly depend on beta cell graft, and here there's not much we can do except that with the promise of potentially in the future of autologous stem cell derived beta cells, antigen-specific therapy would be excellent approach to be combined to protect those autologous stem cell derived beta cells. So if we look at therapies overall in type 1 diabetes, here the use of autoantigen is a small aspect of it. There's a lot of these are not really specific using antigens, but there are also a number of cell therapies that we can consider antigen-specific, like antigen-specific Tregs, or antigen-presenting tolerogenic dendritic cells, which I mentioned a little bit earlier. And these are just examples of how the Tregs or CAR Tregs are already considered by a number of companies in the field. So I want to finish a little bit by saying a few things about what we do in my lab. One large aspect is studying what's happening in human type 1 diabetes, but in an in vivo environment. And for this, we use mice that are reconstituted with a human immune system. So think of it like it as an empty mouse filled with human immune cells, and you can use all sorts of cells, including beta cells, the source that is beta cell that you can graft, and look at how immune cells are behaving and potentially infiltrating beta cell graft, and what are the interactions between beta cells and T cells in an in vivo environment, and also how the immune system in general respond to the types of treatments that I've told you about, particularly antigen-specific T cells. So we can, for example, engineer T cells to express T cell receptors that are specific to particular antigens and look at these particular antigen-specific T cells. And then another major area of research in my lab is to develop antigen-specific therapy for type 1 diabetes, particularly those. So we have a number of platforms that we investigate and developing for the delivery of antigens, and models as well. And I've told you a little bit about it earlier. I just want to mention the peoples I'm working with at Columbia, as well as my external collaborators in all the research we do in the lab. And finally, the funders of our studies, the NIH Breakthrough T1D in particular.
Great. Thank you, Dr. Creusot, for that amazing presentation. Unfortunately, due to time constraints, we will be unable to have our Q&A for Dr. Creusot, and we'll have to move on to our next speaker. If you have any questions for Dr. Creusot, please email us, and we will forward them to him. Arjen will be introducing our next speaker.
Thank you, Dr. Creusot, for your amazing presentation on antigen-specific tolerance and type 1 diabetes. I'm Arjen Midda, and I'm the Vice President and co-founder of Seeking a Path Forward for Diabetes, or SCD. Now, our next diabetes biology speaker received his bachelor's and master's degree from Brooklyn College and his PhD from the University of Illinois at Urbana College. He then was a postdoctoral fellow under Dr. Michael Check at the University of Massachusetts Medical Center and was a professor of physiology and biophysics at the University of Iowa. He became the associate director of the University of Iowa Diabetes Center in 1991 and received the Eli Lilly Outstanding Investigator Award from the American Diabetes Association in 1996 and the University of Iowa Foundation Distinguished Professorship in 2001. And in 2003, he was named the chair of the Departmental of Pharmacological Sciences at Stony Brook University, where he held the William and Jane Knapp Professional Chair of Pharmacological Sciences. So Dr. Pessin came to Einstein in 2008. He is an endocrinology medicine professor, molecular pharmacology professor, holds the Judy R. Alfred and A. Rosenberg Professional Chair in Diabetes Research, and is a director of Diabetes Research Center at the Albert Einstein College of Medicine. So his laboratory focuses on the regulation of insulin and nutrient signaling in the regulation of liver gluconeogenesis and lipogenesis and their dysregulation states of insulin resistance. And today he'll be discussing temporal and spatial regulation of gluconeogenesis in the liver. So without further ado, it is an honor to welcome Dr. Jeffrey Pessin.
Yeah, thank you very much for that nice introduction. Just make sure, can you see my screen? Yep, okay. So I'm going to do, let me give you a very nice talk about autoimmunity and type 1 diabetes. I'm going to talk more about type 2 diabetes and more about the metabolic regulation, particularly in the liver. So as you see, I have no disclosure statements. Why liver metabolism is very important for a lot of reasons, and I'll go through some of that. But one of the important things about what's happening today is because of the increase in obesity, metabolic associated liver disease is becoming a major problem worldwide. What used to be called NAFLD is now called MASLD, and many people have this, and I'll explain that in a second, where they have a lot of fat in their liver, and the fat in the liver causes dysregulated metabolism in the liver. But the problem is not so much the dysregulation of metabolism, it's that MASLD drives the formation of MASH, and that's where you have hepatocyte dysfunction, and you get a lot of infiltration and immune reactivity. MASH then can have different states. You can have it with and without fibrosis, and fibrosis is where you get connective tissue deposition. And when fibrosis occurs, now you start having very severe metabolic dysfunction. And that also leads to more advanced fibrosis and eventually cirrhosis and also hepatocellular carcinoma. And that's a major cause of, main cause of liver failure. And the only cure for that is a liver transplantation. So the progression is becoming a very serious problem metabolically and functionally in people's lives. And just to give you sort of a generic understanding of how many people have this, if you're a diabetic, you take 10 diabetics, 6 out of 10 have MASLD. if you look at obese individuals, 9 out of 10 obese individuals have MASLD. So it's very, very prevalent. As you know, about a third of the US population is obese. So you just think about that. It's 100 million people, and if 9 out of 10 of them, that's 90 million people are walking around with MASLD. Now, it's not just obesity that does this, because even in the normal, regular, healthy, quote, healthy population, the numbers are something like 1 out of 4 people have a fatty liver anyway, right? And that has to probably do with our diets. Okay. The other interesting thing about this, which I'm not going to get into, but is that in adults, MASLD is much more prevalent in men than in women. Women tend to be protected, and that's a well-known phenomenon. And it's also true in animal models like mice, which is what I'll be talking about today. Okay. Before I get there, though, I need to tell you something that we felt is very, very important. Since the progression of liver disease, metabolic liver disease, is a very complicated process, we felt that you have to understand normal physiology before you can really understand pathophysiology. And this was a concept we decided on about 5 or 10 years ago. And we've been very focused now on primarily looking at normal metabolism. And what I'm going to tell you right now is that we don't really understand normal metabolism that well at all. Even though there's a lot known, there's a lot still to be discovered. And because of that, we think that studying pathometabolism is going to be much more difficult to resolve because we don't really understand what's going on normally. So with that in mind, I'm showing you this little schematic here. So normally in humans and in most animals, the fasting glucose levels or the normal glucose levels are in the 5 to 5.5 millimolar range. Okay, that's where you medically it's 80 to 100 milligram percent. Okay, so that's shown here. Now, when you eat a meal, most of you ingested food are mostly fats, they're proteins, and they're glucose. Mostly the glucose you absorb is in the form of starch, except today we eat a lot of sugar. It's either sucrose or it's high fructose corn syrup, or sometimes it's just straight glucose mixed into foods. When glucose levels now rise in the blood because of that ingested glucose, your pancreas, as Remi was talking about, and beta cells start secreting insulin. That insulin goes into the circulation and activates adipocytes and skeletal muscle to take up glucose. At the same time, that insulin suppresses the liver because the liver stores glycogen, another form of starch, and insulin then prevents glycogen from breaking down or the liver from releasing glucose. At the same time, it inhibits new glucose synthesis or gluconeogenesis, and the liver is one of the major organs for synthesizing glucose from precursors. So that drives the glucose levels down, both because the liver is not secreting glucose and because peripheral tissue like fat and muscle are taking up the glucose. And that brings you back to normal glycemia. When you're fasting, you have none of this nutrients coming in. So your glucose levels would start to fall, right? But they don't, or they stay within the normal range because organs like the liver will start synthesizing glucose through gluconeogenesis and breaking down the glycogen that they stored up here, breaking it down and releasing it into the blood. At the same time, this occurs because it's not the beta cells are inhibited from secreting insulin when you're fasting, but your other hormones like glucagon and also epinephrine, which comes from the adrenal gland, will stimulate these more synthetic processes to make glucose. And so now the alpha cells secrete glucagon, that stimulates gluconeogenesis and activates glycogenolysis, again bringing back you to normal. Okay, so how do we study this? Well, we decided to, because we use our experimental models mostly are in mice, we needed a way to control the fasting feeding states. Mice are a little bit different than people, is that mice are nocturnal. They're awake at our night and they sleep during the day. But in an animal room, you can control the mice. You can control when they eat, you can control the light-dark cycle. So what we decided to do is we set up a program where we would feed the mice for 8 hours right? During the light, what we call, I guess, feeding during the light cycle. And then when they're in the dark, they would be fasted. So they would eat 8 hours and they would fast for 16 hours. Then they would eat again for 8 hours and fast for 16 hours. We did this for 3 consecutive days. This makes the mice very hungry, so that when you give them food, they gobble all the food down, saturate their stomachs with whatever food you're giving them. And so they're in a fully fed state. And we found that, for example, lipogenesis gets activated maximally 4 hours after feeding. So what we've done here is, by setting up this entrainment, this controlled or time-restricted feeding, if you like, of the mice, we have very timed mice that are all eating at the same time, approximately the same amount. And so it goes through this various periods. So what we call the 0 hour is when they're fully fed, or no fasting, 0 time of fasting. Now we can remove the food and they go into fasting for 4 hours or 8 hours or 16, 24, whatever time we want, because we have now good control over their ability to eat food and when they eat it. Okay, so this is basically the scheme. They have food access, and I'm just going to show you, I'm not going to show you many time courses, but I'm going to show you what happens in the fasted and the fed state. So the fed state is this time-restricted feeding where they've eaten all the food after 4 hours, the stomachs are fully saturated, and then 24 hours when there's no more food left in their intestinal tract. Okay, so if you look at gene expression, these are typical lipogenic genes involved in lipid synthesis. It's not important what they are, but these are just canonical lipid synthesizing genes. Just to let you know, FASN, this is fatty acid synthetase. That's the enzyme that actually makes the fatty acids. This is an enzyme that elongates those fatty acids. This is an enzyme that provides some of the substrates for FASN to work on. Okay. So you can see here in the fed state, these lipogenic genes are on. So de novo lipogenesis, lipid synthesis, is very high in the fed state. You can see the genes here, okay? But when you fast, these get turned off. The mRNA goes way, way down. And for example, the fold difference between the fasted and the fed state for the lipogenic gene FASN is about 20-fold, okay? Now, if you look at gluconeogenic genes, those involved in glucose production, the canonical gene is PCK1, which stands for phosphoenolpyruvate carboxykinase, is the protein. This is the gene name. But this is a rate-limiting step in gluconeogenesis, glucose synthesis. So in the fed state, the mRNA is mostly down, and in the fasted state, it goes up, also on the order of about 20-fold. So you have this very dynamic range, inverse dynamic range between lipogenic genes and gluconeogenic genes. And this is another one, glucose-6-phosphatase, the catalytic subunit. Okay. So one way you can measure gluconeogenesis, which is what I'm going to focus on, but I'm going to contrast it with lipogenesis a little bit, is that you can do tracer studies. This is a metabolomics type of approach where you do something they call it metabolic flux. There are many different precursors for glucose, and one central precursor is a molecule called pyruvate. It's a 3-carbon substrate, okay? And what you can do is use what's called stable isotopes. Those are heavy isotopes labeled in carbon. So this, instead of being C12 carbon, it's C13 carbon. It's non-radioactive, so it's perfectly safe, but it changes the mass by 1 proton. And in this case, we labeled 2 of the carbons, 2 of the protons in those carbons, so that the mass of pyruvate over the normal pyruvate is 2 atomic mass units heavier. When this gets metabolized into glucose, it can produce 2 carbons in glucose, which are also heavier. And so you can measure all this in a mass spectrometer. So you can measure how much of the unlabeled pyruvate, which is circular in the animal, into unlabeled glucose, and you can measure how much of the stable isotope labeled pyruvate gets incorporated in glucose. So this would be the rate of glucose synthesis, because normal unlabeled glucose can come from many places. It can come from glycogen, as I mentioned. Okay. So this is sort of the enzymatic pathway. I'm not going to go through it. If you guys remember your biochemistry and your enzymatic pathways, you'll remember some of this. If you don't, it's not important. But what I'm showing you is that the glucose is made, here's the pyruvate. In this case, it's stable isotope labeled. It goes like this, like this, like this, back up the glycolytic pathway to form glucose. So if the glucose has these two labeled pyruvate, heavy labeled carbons in it, then that glucose is from de novo lipogenesis, because here's the PepsiK gene enzyme. Okay? All right. So this is the gene expression, fed, fasted. Here's the glucose production, glucose synthesis, gluconeogenesis. So we take isolated hepatocytes, primary isolated hepatocytes from the fed and the fasted state. And you can see in the fasted state, you get about 50% of the glucose is labeled with these two carbons. Okay, that means this is called a fractional synthetic rate, but what it means is that of all the glucoses made, half of them have this two label from pyruvate in them. But because we know gluconeogenesis is higher in the fasted state. What is surprising is that in the fed state, there is gluconeogenesis, and it turns out to be about 12%. So in the fed state, gluconeogenesis is supposed to be off, but in fact, it's not. Okay, so let's see. I only have a couple of minutes left. All right. So let me go through this very quickly. The liver, hepatocytes in the liver are not all the same. Hepatocytes in the liver have different functions depending on their location. There's something called periportal hepatocytes, which are near the portal vein. This is where insulin and factors come in. Nutrients mostly come in this way, and they percolate through what's called the lobule of the liver. And this lobule is made up of different hepatocytes lining the blood flow, the sinusoids, which is where the blood vessels are, to the central vein, where it goes back to the circulation. And so this is oxygen and nutrient rich here. The portal artery is here. And this is oxygen and nutrient poor over here. And these hepatocytes across the lobule behave differently. Okay, and there are different processes. Lipogenesis is mostly over here, and gluconeogenesis is mostly over here. Okay, so what I'm going to show you is, and I'm going to have to do this a little bit quickly. This is what's called single-cell RNA-seq. This is where you can look at how individual cells are actually, what mRNAs they're making. And what we can do is we have markers for pericentral and periportal hepatocytes, and we can just find what they are in a single cell seq analysis. So each of these dots here represents an hepatocyte, and these blue dots are the pericentral hepatocytes. These are the periportal hepatocytes, and these are the hepatocytes that are sort of in the middle. And this is what shows you what it looks like when you do a fasting and feeding. Here's the fasted state. I'm sorry, here's the fed state, and here's the fasted state. And you can see that there's very big differences between the clusters. That means these hepatocytes are very different than these hepatocytes. Okay. And what I'm going to show you is that in the fasted state, I'm sorry, excuse me, in the fed state, there is a subset of these periportal hepatocytes that are making PCK1 or gluconeogenic. And they're right here. So it's a subset. And you can show that if you look in the fed state, that these are all lipogenic. Here's FASN. So there's a subset of cells that are both gluconeogenic and lipogenic, and they're shown here in these red dots. So cells are very, very unusual because they're expressing both gluconeogenic genes and lipogenic genes at the same time. Okay. Okay. Now, the last thing I'm going to show you, because I'm going to skip the rest of the data, is that you can also visualize this spatially by using something called single molecule FISH. It's just like immunofluorescence, where you use an antibody. In this case, you're using mRNA probes. And you can use those probes to look where transcription is going on in the nucleus, where mRNAs are being made. And in the fed state, you can see if you look at PCK1, there are very few cells that are pink, right? These are the gluconeogenic cells, okay? But there are some. If you blow it up, and you look at the nuclei, you can see little dots inside the nucleus. These are sites of transcription. These are transcription units. Now you'll notice, for example, in this one, there's 1, 2, 3. And you would say, well, that's not possible. There must be something wrong because cells are diploid. They only have 2 copies. How could you have 3? Turns out hepatocytes are polyploid. They have multiple alleles, and they can have up to 15 or 20 alleles. And I'm going to show you an example of that. Because when you go to the fasted state, now you have a lot of gene expression going on. You can see lots of pink in the cytoplasm, that's the circulating mRNA, and you can see lots of these little dots in the nucleus, which are the transcription sites. And here you can see there's one cell that has a whole bunch of them, it has maybe 10. You can see them all here. And this is a polyallelic cell. It has multiple copies of this gene, and so it's making a lot of it. But also you can see that it's expressing throughout the lobule, whether it's in the periportal region or the pericentral region. Okay, I'm going to skip this. Okay, and I'm going to skip this and this. And I'm just going to show you this chart. So here is the glycolysis pathway, which is reversible. Most of the steps are reversible except for this one, with pyruvate and phosphoenolpyruvate. Again, it's not important. But for gluconeogenesis to occur, the pyruvate has to get into the mitochondria. It has to go through the TCA cycle, back into the cytosol, where this key enzyme, PepCK, will convert phosphoenolpyruvate back to precursors that can go up the glycolytic pathway. On the other hand, to make fatty acids, pyruvate has to go to acetyl-CoA, so it can get out to citrate, to get out into the cytoplasm, and then it can go and undergo de novo lipogenesis through this key enzyme, fatty acid synthase, which I showed you before. So how can glucose go down this way to make fatty acids, at the same time go this way to make glucose? This is a conundrum, but that's what the data is telling us. So it's telling us we don't quite understand how normal physiology is working because we have cells that seem to defy the normal way we think of enzymology is supposed to work. And that's one of the main things our lab works on, is trying to figure out how this can actually happen. Okay, so with that, I thank you for your attention. And if there's time, I'll answer questions. If not, we'll wait till later.
Yep. Thank you, Dr. Pessin, for that amazing presentation. And yeah, just to be on schedule, we're going to be moving all of our Q&As to the Zoom chat. So if any of the attendees have any questions about the presentations, please place them in the chat and then the speakers will respond to you. And if the speakers are unable to respond in the chat, we will email the speakers those questions. And furthermore, you can ask the speakers your questions in the breakout rooms. So now Avi will be introducing our next speaker.
Thank you, Dr. Pessin, for your presentation on gluconeogenesis in the liver. I'm Avi Pandit, treasurer of SED. Our next 2 speakers will be discussing diabetes population health, how diabetes affects people, analyzing it at the more macro level. Our next speaker earned her MD degree cum laude from the University of Toronto and a Master of Science in Clinical Research Methods cum laude from the Albert Einstein College of Medicine. She has served as an associate editor of the American Journal of Physiology and secretary-treasurer of the American Federation of Medical Research. Dr. Hawkins is a Basina Scholar of the American Federation of Aging Research and has received research grants from the National Institutes of Health since 1999, the American Diabetes Association, the Juvenile Diabetes Foundation International, and the Diabetes Action Foundation International. She is the recipient of numerous honors and awards, including the Junior Physician Scientist Award in 2001 and the Outstanding Investigator Award in 2012 from the American Federation of Medical Research, the Novartis Young Investigator Award in Diabetes Research in 2007, and election to the American Society for Clinical Investigation in 2012. She recently served on the congressionally appointed National Clinical Care Commission. She is a professor of medicine and associate director of the Einstein Mount Sinai Regional Diabetes Research Center. She also holds Harold and Muriel Block Chair in Medicine. Lastly, she founded and directs the Einstein College of Medicine's Global Diabetes Institute with the goal of building strategic partnerships in medical research and education to combat the burgeoning global diabetes epidemic. Today, she will be discussing welcoming type 5 diabetes, a global paradigm shift. Without further ado, it is an honor to welcome Dr. Meredith Hawkins.
Thank you, Avi, and I want to very warmly thank the organizing committee. I think you have tremendous vision in setting up and organizing what's taking place today, what we're taking part in. It's so badly needed, and the idea of involving students in order to get excited about this area. This is just, as I'm going to show you in a second, this is a burgeoning area globally, and it's our pleasure to give talks that really span the spectrum from basic science to population health. And mine will look at population health. As I show you this slide, it does show you, in fact, one of our global partnerships. So between Einstein, our institution, and Christian Medical College in Vellore, India, very renowned institution, whose symbol is just below ours, this has been the result of about a 15-year collaboration that I'll show you today. And this is the late-breaking news. You heard it here first. Have any of you ever heard of type 5 diabetes? Well, you're hearing about it now, and the rest of the world will be hearing about it on April 8th, when it will be proclaimed to be such by the president of the International Diabetes Federation. So I'll be leading up to how that came about. First of all, I want to show you why I'm so glad you're all here, and that is because even compared to other serious diseases, if we think about HIV/AIDS having a global prevalence of about 35 million, the current estimated prevalence, based on an article last year in Lancet, current estimated prevalence is 730 million people with diabetes globally. You can see that this slide from the IDF hasn't even quite caught up with that yet, but what you'll see with these upward arrows is that we know that it's increasing all around the world. And so the world on the upper left, you'll see we're projecting between 2021 and 2045, projecting a 46% increase. As far as the numbers, we've already exceeded them, as I just mentioned. But you look at where else in the world it's increasing massively, and two other areas of note would be Africa, with a projected 134% increase over that time period, and Southeast Asia, 68% increase. How could it be that areas of the world that we know are associated with undernutrition could be seeing these mass increases in diabetes? Well, not everybody in these countries is undernourished. A number of them would be living in the slums where they could be buying unhealthy food and actually managing to become overweight and developing diabetes. But that's not everybody. We're still in these countries, still seeing a massive number of people who are underweight. And our interest over the years has been in the diabetes that manifests in these individuals. I'm going to give you a little bit of a history of something that was once called malnutrition-related diabetes. This entity was first described 70 years ago, reported in Jamaica in a landmark Lancet paper, described as J-type diabetes because that's where it was reported. But soon thereafter, it was noted that this form of diabetes was popping up all over the world, tropical countries. I'll show you a map. The characteristics of this, as I'll expand upon in a minute, is that these are malnourished individuals who have a type of diabetes that seems to defy the definitions of type 1 and type 2. It's non-autoimmune. They don't go into ketoacidosis. They're also not overweight. And I'll show you a little bit more about exactly what does constitute this entity. Now, in 1985, the World Health Organization, acting on many reports that had been published around the world by that time, decided that this entity warranted a classification. So it was called malnutrition-related diabetes. Unfortunately, in their 1999 diabetes classification, it was removed. They cited that there was a lack of convincing evidence of causal association with malnutrition. It was after that time that I personally started to encounter a lot of these individuals. Some of them I encountered in person when I was working in the wards of a big hospital in Uganda, and many others I heard about when I went to speak at various global health meetings. And I was approached by physicians serving all around the world in low and middle income countries who kept telling me the same thing. They were seeing a lot of very thin patients with diabetes, and they wondered what they were. So as noted in the middle bar here, various reports have come out of many different countries. I'll show you all on a map. But we believe now that there's probably about 25 million people affected globally with this form of diabetes. But, as you'll see in the next point, because of the lack of formal recognition, this form of diabetes has been understudied and misdiagnosed. In 2022, responding to that call for more research, we published what amounted to be about a 10-year research project that we conducted in partnership with our colleagues in India. I'm going to go through that with you, exactly what we did and what we found. But the bottom line is we now have had over 12,600 reads of this paper. Those are people from all over the world reading about it, and that has really started to change the landscape, as I'll tell you. But I'm going to tell you a little bit about what we did in this research project that is hopefully starting to change the field a bit. Now, what was known before we did our work was all from case reports or case series. And the prevailing characteristics that were being reported was that these people tended to be young, they tended to be lean. Their history was that they had poor nutrition in utero, so these people were being born with low birth weight. But the problem was that the malnutrition continued. These were not people who had just survived a famine and then were being refed. The individuals that we met when we met them as adults were stunted in height, were seriously malnourished. They had reduced lean body mass. They had low overall BMI, and they had basically never caught up in terms of their nutrition status. When they presented in small rural clinics with diabetes, it tended to be severe. And for that reason, these individuals were always placed on a lot of insulin. And so the word on the street, as it were, is that these people were insulin resistant because of the amounts of insulin that was being prescribed to them. However, they did not go into ketoacidosis, but what in fact unfortunately happened more often than not when we saw them in Uganda was that they were treated as though they had type 1 diabetes. They were given insulin, quite a bit of insulin because their glucose levels were high, sent off to their village without adequate means of checking their blood glucose levels, and then also without adequate food. And sadly, virtually all of these young people who we met in Uganda and who we hooked up with help and supplies and insulin, so many of them died of low blood sugar. That became a galvanizing call to action for us to begin a project. In order to remember individuals like Ponciano, shown on the right, one such individual, the son of a widow, finally got medical care 4 months later, died in the middle of the night. After he'd been doing so well, he then succumbed to hypoglycemia, showing that by not knowing enough about this condition, these impoverished individuals were getting the wrong care and frequently dying from their treatment. Now, as I promised I'd show you a map, and you'll see that the reports of this condition have been around the world, but around the tropical regions and low and middle income countries all around the world. We opted to perform some, what we say, state-of-the-art research studies in Vellore, India, which became our partner institution. And that was because they had the capacity. They had access, if we went far enough out into the countryside, they had access to individuals with this condition, but they also had state-of-the-art methodologies that would allow us to study them as intensively as we could back in the US. So on the left, you can see a 3 Tesla MRI so that we could actually look at fat depots all through their body. At the bottom, you can see indirect calorimetry to look at energy expenditure. And then on the lower right, you can see the type of a study that we and only a few other institutions do. It's considered to be the gold standard for studying metabolism. It's what we call an insulin clamp study, in essence, an infusion study of insulin and other hormones, keeping all parameters very fixed, and being able then to carefully measure liver and peripheral glucose metabolism. So whole body glucose metabolism can then be compared individual to individual. And so we went about studying 5 different groups of individuals. The first, it took many years to locate these individuals. These were the individuals with low BMI diabetes. We had to very, very carefully exclude all other known forms of diabetes. So in order to get to this number, we had to exclude monogenic diabetes, any antibodies, any evidence of type 1, really just about any other known form of diabetes that you can think of, hereditary lipodystrophy, etc., to get down by exclusion to this group that really seemed to be the same individuals that had previously been reported as malnutrition-related diabetes. And as you'll see there, A1C levels were much higher than the other groups of diabetes. We had a group with type 2 diabetes, and although they were relatively lean when we think about type 2 diabetes, we had to have a second group of controls just to match them because they were different in BMI from our lean, as we would expect and hope. They differed from our low BMI group. We then studied a group with type 1 diabetes, then a group of lean non-diabetes individuals who were again matched by BMI to the low BMI group, and then a group of what they termed overweight non-diabetic individuals who were the controls for the type 2s. Now, I'm going to show you 3 result slides. First one, during those clamp studies where we infused basal insulin, we first looked at what we call endogenous glucose production, primarily the amount of glucose that's being produced by the liver. And the more sensitive somebody is to insulin, as you can imagine, the less glucose production they will make, as Dr. Pessin so elegantly showed. So if you are then insulin sensitive, like the individuals in green, who were the lean non-diabetic group, you'll see that they suppress their glucose production in the presence of quite low insulin levels. The overweight individuals to the right in the gray circles, they were a bit more insulin resistant. As we might expect, they were overweight. But the ones who were the most insulin resistant were the ones in the middle bar. These individuals had type 2 diabetes. As we would predict, their livers were less sensitive to insulin, so their glucose production was higher with the same levels of insulin. And intriguingly, the type 1 diabetes trended towards insulin resistance as well, as has been reported in the literature with long-standing type 1, whether it is that chronic exogenous bombardment with insulin, but they showed some degree of insulin resistance, not to the same degree as type 2. But you'll see on the far left, these were the individuals with low BMI diabetes. And although there was some heterogeneity, you'll see that most of them clustered around here, showing that they were very insulin sensitive, and they did not differ statistically from the non-diabetic individuals, indicating that the level of the liver, these individuals were insulin sensitive, unlike the literature. And now I'm going to move from the liver to the rest of the body, whole body. A lot of that involves glucose uptake into skeletal muscle, into fat and other tissues, anything under the influence of insulin. And as a comparison, we're going to start with the normals, the green circles here towards the right. And you'll see that when you're more insulin sensitive in these tissues, you have more glucose uptake. So these are higher levels of glucose uptake, a little bit diminished in the overweight group on the right. But in the middle, the type 2 group, just as their livers were insulin resistant, so their other tissues are insulin resistant. So that with the same amount of insulin infusion rates, what we saw here was that they were much more insulin resistant. They had much lower rates of glucose uptake. The type 1s are here in the salmon color, fairly close to normal. And then on the left, we see the individuals with low BMI diabetes, and they did not differ, though being a bit heterogeneous, but overall did not differ from the normal subjects, indicating once again, this is a big paradigm shift from the literature, but indicating that if you looked at them carefully, if you first corrected their glucose levels and then studied them carefully, they were not, in fact, insulin resistant. They were just as sensitive as normal individuals. So why then do these individuals present with such severe diabetes? The next part of the study was to look at insulin secretion, giving them a mixed meal drink, and then studying them for 3 hours later, measuring C-peptide levels and using a mathematical model to generate these curves to look at insulin secretion. Normal is in green. So these non-diabetic individuals showed a very nice peak, as one would expect. That's called the first phase insulin secretion, up to 60 minutes, and then it drops off again very nicely. The groups that were overweight had a slightly heightened insulin secretion response, probably because they were veering towards some insulin resistance. But by comparison, the group with type 2 diabetes in black showed exactly what we would expect. Their insulin secretion rates were reduced. You'll see a marked reduction in the first phase of insulin secretion. They only make it about halfway up to normal, and then they just don't come back down again. So they keep circulating higher levels of insulin, which may in turn have detrimental effects. And then type 1 in the salmon, although they could detect with a very specific assay, could detect tiny amounts of C-peptide, that did not respond to the meal. So you can see that they stayed down here. And how about our individuals with low BMI diabetes? You'll see them here in blue. It was quite striking. They were not as severe as type 1 diabetes, but they showed a degree of insulin secretion defect that was more severe than type 2. And that really is saying something, because we know that to develop type 2 diabetes, you have to have an insulin secretion defect. So you'll just see to what extent they were reduced even lower than those with type 2 diabetes. They had just enough insulin to prevent the production of ketones, but otherwise markedly reduced insulin secretion. So to conclude, this is the first study to use carefully controlled conditions to study this atypical low BMI diabetes that had lacked a name for so long. And we showed that they had markedly decreased insulin secretion, but overall no significant insulin resistance, suggesting that this is a distinct entity deserving its own classification. And that this would have massive implications for treatment. I'll just go over this quickly, but as I noted, agents that precipitate hypoglycemia are dangerous. We might need to look more at high protein diets and other approaches that are specific for this entity. So what's in a name? As Shakespeare said, well, lacking a name can be very problematic. This led us to bring together many people that have been studying this on the ground without much fanfare for many years in places like Ethiopia, Rwanda, Uganda, India, Bangladesh, and we brought them all together to a meeting, a consensus meeting in early January. And the idea was then that these people would present this to experts in the field, including leaders of several major diabetes organizations globally. And at the bottom, you'll see that they unanimously voted that a distinct non-autoimmune diabetes in undernourished individuals warranted its own classification as type 5 diabetes. This will soon be published in the Lancet Global Health. This is a picture of all the attendees and a picture of Michael Schwartz, president of the International Diabetes Federation, who will be declaring this on April 8th, that this will be in fact its own classification. And this is the anticipatory slide of our inaugural event. And I wish to thank all of the people whose very hard work made this all possible. Thank you.
Cool. Thank you so much, Dr. Hawkins, for that amazing presentation. As a reminder, our Q&As are now in the Zoom chat, just in the interest of time. But now let me just introduce our next speaker. So first of all, I'm Ashwin Hariharan. I'm one of the co-directors of local involvement here at SCD. And our next speaker earned her higher diploma in French at the University of Bordeaux-Montaigne and her MD degree at Universidad Veracruzana. She completed her residency here at the University of Southern California and her fellowship under the Division of Metabolism and Endocrinology at the University of Washington School of Medicine. She's currently a board-certified endocrinologist and associate professor at the University of Washington School of Medicine, and as a clinical director of the groundbreaking Latinx Diabetes Clinic at UW's Medicine Diabetes Institute, she's dedicated to dismantling health disparities through culturally and linguistically tailored healthcare. Her clinical research, including her 2018 Latino Center for Health grant-funded pilot project, Compañeros en Salud, focuses on improving diabetes self-management within underserved populations. Beyond her clinical and research work, she's a strong advocate for diversity in medicine, serving as chair of Equity, Diversity, and Inclusion at UW's Division of Metabolism, Endocrinology, and Nutrition. Today, she will be sharing her insights on addressing disparities in diabetes care. Without further ado, it's my honor to welcome Dr. Lorena Wright.
Thank you. Good morning, and thank you so much for the invitation. Can you hear me okay? Yeah, okay, good. Yeah, I'm having a little bit of problems with the Wi-Fi, but I think I figured it out. Hopefully we'll be okay. Yes, and thank you so much for inviting me. I was asked to talk about my work at as the director of the Latinx Diabetes Clinic. So I want to share my, can I share my screen or?
Yeah, you can share your screen.
Okay, good. So let's see. I just, Share. There we go. Okay. Okay, great. Yes. So I will then, talk about addressing disparities in diabetes care. And thank you so much for the nice introduction. Yes, my name is Lorena, and I'm the director and founder of the Latinx Diabetes Clinic at the University of Washington in Seattle. So I'll tell you a little bit about this clinic and the work that we are doing regarding healthcare equity at the University of Washington. And as you know, diabetes affects minoritized groups. And here, I'm showing you a graph. I'm not going to give you a lot of statistics, but I just wanted to show you a graph, of the ethnic groups that are most affected by diabetes. And as you can see, the prevalence of diabetes is the highest in Native American individuals, followed by Black individuals and Latino individuals. But even within the Latino group, diabetes affects more of certain groups of Latinos. We are not all the same. And Mexicans are most affected, followed by, Puerto Ricans. And here, like Asian Indian individuals are also re-affected in Black and non-Hispanic people. Not only, we are most affected by or frequently affected by diabetes, but that once we are diagnosed with diabetes, we are diagnosed at younger ages. And also, once we are diagnosed with diabetes, we tend to have more complications and also mortality is significantly higher. So Mexican Americans suffer about 70% more kidney failures as a consequence of poor diabetes control compared to non-Hispanic whites. We have 50% higher risk of dying from diabetes, and also amputations, lower extremity amputations. The rates are higher in African Americans and Hispanic patients. So, and like when I became an endocrinologist, then I became more interested in diabetes. I always wonder why that was, right? Like, what are the factors, or why is it that we tend to do worse? And well, this is multifactorial, right? Like, but in part it is because of disparities in healthcare. And I really kind of like focus on this because I feel that these are things that we can do, really, that we can do to decrease these healthcare disparities and decrease the rate of complications, decrease mortality related to diabetes. But this is, like a study or a survey that look at, the standards of care for diabetes. As you know, like it is a standard of care to measure A1Cs, to do feed exams, to do dilated, eye exams, to manage cholesterol as part of diabetes care. And you can see that Hispanics or Latinos, they had less A1Cs done, less feed exams, less eye exams, and less cholesterol tests. And these, these models were adjusted for insurance because, of course, like if someone doesn't have insurance or doesn't have a primary care provider, then these services are not going to be done as frequently. But this was actually adjusted for insurance, for poverty level, education. And despite all these adjustments, disparities still persisted, for Hispanics, Blacks, and Asians. So, and food exams for Hispanics. So then, not surprisingly, diabetes outcomes, at least in Latinx diabetes patients, are partially due to healthcare inequities. And that will include inadequate access to culturally sensitive healthcare, to language appropriate care, and so on. So then that's why, I felt that, at the University of Washington, we had like the resources. So it was just a matter of kind of like organizing them and have this initiative that would address some of these disparities. And so then we officially inaugurated the Latinx Diabetes Clinic in November of 2020. I had, been doing a lot of this work already, just like the clinic didn't really have this like official, like name. And this is my colleague and friend, Dr. Morales. We both stations at the Latinx Diabetes Clinic. And I will tell you a little bit, I'll tell you more about the work that we are doing at the Latinx Diabetes Clinic, because I'm hoping that, well, not only, like we aim to reduce healthcare disparities, but there we also have other, other objectives. So the mission of our Latinx Diabetes Clinic is kind of like concentrated in 3 bullet points. One is to decrease diabetes disparities by addressing linguistic and cultural barriers. Number 2 is to enhance the teaching mission of the University of Washington School of Medicine, specifically when it comes to cultural and sociological aspects of diabetes care. We also aim at improving the overall health of the Latinx community through increased clinical research. So then I'll tell you a little bit more about like each of these bullet points. So the first one, like to decrease diabetes disparities by addressing cultural and linguistic barriers. So we officially inaugurated our clinic here, like is with part of the staff. This was in 2020, so in the middle of the COVID-19 pandemic. So then at the time we couldn't even take a picture without our masks. And I have to say that during the COVID-19 pandemic, these healthcare disparities became even more obvious, especially in minoritized groups in Latinx community. It was significantly affected by COVID-19 in the state of Washington, and I think nationwide. So in our clinic, we aim at providing bilingual care. We have our nutritionist who is bilingual, or a staff at the front desk, help patients with referrals and follow-up appointments and so on is also bilingual. Or diabetes educators provide, the education in either Spanish or English, whatever language the patients prefer. We also aim at increasing access to technology and new therapies. It has been shown that there are significant disparities when it comes to continuous glucose monitor use and insulin pump use. So we aim at decreasing those barriers, like that if a patient doesn't really want to start an insulin pump or a continuous glucose monitor, that will be because if there is their choice, but not because that this option has not been offered to them. We also offer referral to specialized care, so nephrology, cardiology, ophthalmology, vascular surgery, like whatever the patient needs. If they are having acute kidney failure, chronic kidney disease, well then they are referred also to the nephrology clinic. And very importantly, we also provide financial assistance. So then if the patient doesn't have insurance, then we provide financial assistance. The patients need to fill out an application and so on. So then that is really not like a barrier. In addition to having this clinic, specialized clinic, we also do a lot of outreach efforts. We have a lot of outreach. Here with Dr. Morales, part of our staff of the Latinx Diabetes Clinic. So we go to healthcare fairs and so on, because we try to reach people who might not know about our clinic or our services, and we participate, like in events, like here, for example, I'm here like in a Zumba class in the community here. I'm with part of the staff as well, right? So then the goal is to reach populations that might actually not know about our services, and during these health fairs, we screen for diabetes, we screen for hypertension, for dyslipidemia, obesity, and so on. But we try kind of like to make this, a cultural event to reach these communities. The second bullet point of the mission of our clinic is to enhance the teaching mission of the University of Washington School of Medicine. So we have clinical rotations for internal medicine residents and endocrinology fellows is a mandatory rotation. We also have opportunities for quality improvement projects for trainees. And just last year, we also started in collaboration with the University of Washington School of Social Work, a practicum for bilingual students at the School of Social Work. And this is Michaela. She is presenting, her work in our Latinx diabetes clinic. She was the first of our social work students in our clinic. So as I mentioned, we have like a lot of opportunities for trainees. Really, this is Jane. She's an endocrinology fellow. She look at disparities in telemedicine use in our clinic, like to show that if we don't provide, telemedicine services in Spanish, or like the platforms are not, like in Spanish, then it's going to be a significant disparity. So then we are working on improving that. Arita, she was at the time an internal medicine resident. She's now an endocrinology fellow at Stanford. She showed, that the use of CGM in Latinx patients with diabetes improves A1C, and this technology was very, very well received. And all these, studies, they are very important because then they are, this is data that will help shape policies. So then, like we aim at having CGMs more accessible to patients and so on, and telemedicine services, so on. So we also provide observerships for talented international bilingual medical, doctors, who, and I'm very proud, of these doctors, because they, they rotated in our clinic. We help them improve their chances to match in a residency program in the US. So Lucia is currently like a second-year internal medicine resident at the University of Washington. Abraham is now like her first year. Lucas and Juan, they had just matched, and they will start their residency program, hopefully. They will start, I think, in July. So the third bullet point is like to improve the overall health of the Latinx community regionally and nationally through increased clinical research. So, in our clinic, we provide culturally appropriate care, but we also want to serve as a model. So then we have Compañeros en Salud. This is a diabetes program. It is composed of 12 modules, and here are the topics. And this is culturally appropriate for the Latinx community. It's like all this education is kind of like in the form of stories, like all us Latinos, we kind of like love stories, and this is kind of like a fun way to learn about diabetes and healthcare when it comes to diabetes. So we have tested this educational program, and it was very well received. And then later on, then we started this program. This is a randomized controlled trial that uses the Compañeros en Salud program, this diabetes education program, paired with continuous glucose monitor in patients with type 2 diabetes in the community. And the protocol has been published, and the results of our study is in press. So hopefully it will be published soon. Look, I can show you the results because it's still on embargo. It's about to be published, but we presented our findings. An improvement in hemoglobin A1C. The program was very well received, and we aim at having this program, like to spread this program to other clinics and for that to become a model. So then with all the work that we have done in our Latinx diabetes clinic, and increasing teaching and the importance of culturally appropriate care, and our research in the community, we just kind of like want to show that that these interventions hopefully help decrease disparities in diabetes care in the Latinx community and beyond, or like in beyond other populations. So to conclude, I want to leave with with the points that to eliminate the diabetes disparities, we really need to increase access to bilingual but not only bilingual, but also culturally appropriate care. We must improve in this area. We also need more research on new methods of evaluating clinical, behavioral, and psychosocial aspects of health, which is what we try to do with our study. And we need to implement multi-level programs from promoting prevention to for screening for asymptomatic adults and identifications of those individuals who are at the highest risk. Improvements and access to quality medical care to optimize provider and patient knowledge alike. So I leave you with this quote that to eliminate disparities, we must know enough, right? We need to know research. We need to prove that diabetes disparities, is not an opinion. It's not a matter of opinion. The data shows that, right? We need to do enough. We need to deliver the outcomes. We also need to care enough. We need to be committed to our work, and we need to persevere enough. Don't get discouraged. We just need to keep fighting. Yes. And that's with that, I think I will be happy to answer questions, I think, in the chat. Right? Yeah. Thank you so much for listening and your interest.
All right, thanks, Dr. Wright, for those amazing insights. We are now going to be having our networking break, which will go until about 10:45 a.m. PST or 1:45 p.m. EST. During this break, attendees can network with the speakers. There will be 4 breakout rooms with 1 breakout room for each speaker category, so 2 speakers total per breakout room. A member from our organization will be moderating in the breakout room and will display questions that the attendees can potentially ask the speaker and vice versa to facilitate conversation. These questions will be about the profession of the speaker, why the speakers and attendees are interested in diabetes, and less so about the presentations themselves. I will let everyone know when the breakout rooms are about to close by displaying a timer with how many minutes are left in the breakout room. Attendees will be able to hop to different breakout rooms if they want to. So in room 1, we will have our diabetes biology breakout room with just Dr. Pessin, as Dr. Creusot had to leave early. Room 2 will be a diabetes population health with Doctors Hawkins and Wright. Room 3 will be a diabetes clinical care with Doctors Basina and Shih. And room 4 will be diabetes technology with just Dr. Prioleau, as Dr. Doyle had to cancel for today. Okay, so the breakout rooms I'm going to be opening right now. Okay, the breakout rooms should be open.
Hi, I can't join. I'm not sure if I'm pressing right. Okay.
Yep. There should be, do you see like a breakout room button?
I don't.
You do not.
Maybe.
Okay. Oh, open all.
I don't either.
Oh, now I
Okay,
do.
there we go. Okay.
Disappeared.
I
Thank you
don't
so much.
Thank you. Yep.
I don't see it either, so I don't know.
Yeah, it looks like I just opened it. Dr. Wright, are you able to see it?
It just appeared for me at the bottom. I'm not sure if it's for you too as well.
It appeared and then it disappeared. Actually, I saw it for a second pop up at the bottom.
breakout rooms, okay.
Oh, here we are. Let's
Mine
see what
is
happens
too.
Correct.
here. Okay, great.
This is my break is the 2. Sorry.
Correct. Yeah, room 2.
Okay, great. Okay.
Hi, Dr. Prioleau. Yeah, it looks like you're in room 4.
Yeah, okay. I think everyone's back in the main room. So I hope everyone enjoyed getting to network. My name is Tiara Joseph. I'm the Director of Member Engagement at SCD. Our next set of speakers will be discussing their insights on diabetes clinical care, from how to improve diabetes clinical care to their perspective as a diabetes clinician, etc. Our first speaker received her medical degree from Nikolai Pirogov Moscow Medical Institute and her endocrinology fellowship from Stanford. She is a clinical endocrinologist and clinical researcher with a focus on diabetes management, thyroid, and adrenal conditions. Her primary interests are in type 1 diabetes, technology, and diabetes in pregnancy. Dr. Basina is board certified in endocrinology and internal medicine. She received numerous number of medical advisory boards for several community diabetes organizations. She's a medical director of the inpatient diabetes program and chair of the diabetes task force at Stanford Medicine. Today, she will be discussing unraveling diabetes, insights from research and future directions. So without further ado, it's an honor to welcome Dr. Marina Pessin.
Good morning, still good morning in the West Coast. And again, thank you so much for organizing this great event. And thank you for a nice introduction. So I'm going to share my slides. Okay, can you see my slides okay? Great. Okay, so I am as you've heard in the introduction, so I work at Stanford University, and I just have pictures of our beautiful new hospital with the very famous buckyball, as you can see in the middle. So I think there are very few of them in the country. And it does light whoever is coming to Stanford. Just look at that. It just lights up at night, and it's really, really pretty. So I will talk about some of the projects that I am involved in. So one is the DREAM trial. This is a diabetes related to acute pancreatitis and its mechanisms. So acute pancreatitis results from an acute inflammatory injury for the pancreas. It is the most common GI discharge diagnosis in the United States, and it accounts for over 300,000 hospital discharges per year. 30 to 40% of patients develop diabetes or some sort of impaired glucose tolerance or some dysglycemia within about 3 to 4 years of even a single episode of acute pancreatitis. So with that, National Institute of Diabetes and Digestive and Kidney Disease and NIDDK found the type 1 diabetes and acute pancreatitis consortium in order to undertake a prospective longitudinal observational study, sort of creating this cohort of the occurrence of diabetes during an acute pancreatitis episode and subsequently with emphasis on type 1 diabetes. So our study overview is to determine the cumulative incidence and clinical characteristics associated with the development of diabetes after one or more episodes of pancreatitis, characterize beta cell function and endocrine alterations after acute pancreatitis and their relationship with the development of diabetes after acute pancreatitis, and also to determine the immunological mechanisms of diabetes after acute pancreatitis, including the contribution of beta cell autoimmunity. It is a longitudinal study that enrolls individuals over the age of 18 who had experienced one or more episode of pancreatitis. It's a multi-center consortium. It's an observational study that it's still, I think we're in the year 3 or 4 of the consortium, and actually USC participates as well. So there are multiple tests that we are doing for those patients, and our coordinators get paged with each elevated lipase in the hospital, and they determine whether the patient is eligible for being enrolled in that study. So we're trying to look what kind of incidence of type 1 and type 2 diabetes or prediabetes following an acute pancreatitis. What are the clinical risk factors that are present for development of diabetes or prediabetes? And we're looking at different metabolic alterations that happen after an episode of pancreatitis with insulin secretion, beta cell function, and sensitivity to insulin. What are the hormones that are released from other islet cell, such as glucagon, pancreatic polypeptide, that we're measuring during a mixed meal tolerance test that patients are getting, and also release of the incretin hormones. So the other organization that I'm a part of, and I have been a chair of the Data Science Committee. It's a Type 1 Diabetes Exchange. It's a wonderful organization that was found many years ago. And right now it includes 61 institutions throughout the country that provide care of over 150,000 individuals with type 1 diabetes. Type 1 diabetes members, it's a QI collaborative, share data via type 1 diabetes quality improvement platform. So we all, not all, but I would say majority of the institutions, we do what called data mapping. So our de-identified data on type 1 patients, and now we're starting type 2 diabetes data mapping that are being kind of flowing continuously to type 1 diabetes exchange portal. And whoever is in the consortium or in the portal, it has access to all this data. It's all de-identified. And if anyone wants to look at some kind of research question, you can access this data and look at this 150,000 patients to be able to study certain questions, your study questions. This is, of course, aimed to improve outcomes and also measurably improve the lives of individuals with type 1 diabetes. And those are the institutions that are involved from the most recent map that was presented at the collaborative annual meeting that we just had at NIH. So this is just an example of what we do. So we do a lot of different studies looking at the data from the collaborative as well as our own data from our own database. So this is an example of the project that one of the students did and presented at American Diabetes Association in 2024. So the goal for this project was to look at patients who are getting closed loop system with Omnipod 5 insulin delivery system and to see what's the best way of training of those patients and what are the obstacles, what are the different challenges that those patients have and how we can create that best training platform, e-platform, as well as the in-person platform to train those patients. So she came up with a fishbone diagram with the key driver diagram, KDD. So I think this is extremely great projects and participation because you will be doing QI throughout your career. And we get a lot of, through T1T Exchange, we get a lot of coaching on the QI projects with a QI coach who meets with us every 1 to 3 months, guide us sort of on the project and helping the students or whoever the trainees are participating to learn about different QI projects. So other study that I am very involved in that called Spirit Study. So this is actually a great maybe continuation or also a link to autoimmunity work that was presented earlier this morning. So we have a Spirit Study, which stands for Stanford Pancreatic Islet Cell Replacement and Immune Tolerance Initiative. So we have two arms. One is for autologous beta cell transplantation, and the other one is for allogeneic beta cell transplantation. So autologous is for patients who have chronic pancreatitis with a lot of with frequent admission to the hospital and requiring different pain medications, chronic pain medications. And those patients elect to or are recommended or elect to undergo total pancreatectomy. So when they undergo total pancreatectomy, obviously they develop type 1 diabetes. So the program includes, it started in 2009, and it's a multidisciplinary care that involving multiple specialties. We do have about 500 patient database with chronic pancreatitis. So we propose that we will be due 3 to 4 autologous transplantations per year, but actually, so in 2004, we did 4, and we already had done 1 this year, and we have a couple more patients scheduled. So it's quite a interesting, I would say, process and quite labor-consuming process, very well organized, like really, the program is running fairly amazingly well. We do have the allogeneic transplantation program, which is for type 1 diabetes patients with severe hypoglycemia and awareness, who are going to undergo the donor transplantation. So we have, it's not FDA approved, and autologous actually is FDA approved and it's insurance covered, but allogeneic is done as a part of research. So the idea of our program is to create immune tolerance that patients will have islet cell transplantation along with a mini bone marrow cell transplantation to develop that sort of the chimerism protocol to develop this immune tolerance. And the idea is that within 2 to 3 years, patients will be off immunosuppressant medications. So we have a couple patients consented. No one has gone the cell transplant yet. But hoping to get it done soon. So this is just the overview how the islets get isolated. So it's a Dr. Camila Ricordi's chamber, who created from University of Miami, that I had an opportunity to see a few times, go into his lab. So pancreas gets kind of digested, and this is Ricordi's chamber, and pancreatic cells get purified, and they get sort of collected into this basically IV bag, and then they get infused into the portal vein, into the portal circulation by our interventional radiology colleague. So in order to get good results, you need to get at least, ideally, at least 5,000 cells per kilo. But so far for our autologous transplantation, because patients were already pretty far advanced in their pancreatitis and they lost substantial amount of beta cells, we were able to obtain anywhere from 1 to 3,000. And so far, all the patients are still on insulin, but they are doing very well, and they're super happy that they had the procedure done. Other fun study that is currently ongoing, it's called the AIDing study, which stands for Automatic Insulin Delivery for Inpatient with Dysglycemia. So those are participating institutions who are collaborating with Emory and University of Virginia. This is a Omnipod. So the insulin in this pod fits 200 units, stays on the body for 3 days. It talks to the CGM, which is the Dexcom. This is the older version, so I should have changed the picture. So we changed it. Now we're using the G7, the newer version. And this is a PDM, which is a Samsung phone that blocked for all applications except the Dexcom and Omnipod. And you can see, this is the front screen. So it shows that patient, we call this little hamburger, says automated, which means patient are getting automated delivery by the little pod. So the data from the sensor goes directly to the pod. Pod has algorithm, and every 5 minutes, insulin being delivered based on the algorithm. So we are enrolling inpatient from either type 1 or type 2 diabetes requiring insulin therapy. We have been enrolling the most challenging patients who are transplant, who are receiving tons of steroids, who are in tube feeds, TPN. The system so far have been performing extremely well in those patients. This is a randomized controlled trial with one-to-one randomization. So controlled patients are on continuous glucose monitoring alone. And we are looking at efficacy and safety of this automated insulin delivery system versus standard therapy with multiple daily injections. And also looking at certain metrics. So it's a very cool study. So this is our setup. So basically, patients have the remote monitoring. So by the study team, so we are seeing every 5 minutes what's happening with the patient's blood sugar and insulin delivery on, I can see now on the computer. Patients in the room wear the pod and continuous glucose monitoring device. The PDM or the actual controller is locked in the plastic box that's next to the patient's bed. And nurses at the nursing station, they have this iPad, and it's like cardiac monitoring, but they can see what's happening with the patient currently, and they get CGM alerts for hypo and hyperglycemia. So another study that I'm involved in, and I don't know if I'm running out of time, but this is to prevent complications in type 1 diabetes. About 25% of type 1 diabetes individuals develop chronic kidney disease, and lifetime risk of kidney failure is 10 to 30%. Tight glycemic control had been shown to be improving outcomes, but there are other factors that are important. And there's a medication that's called SGLT2 or sodium glucose transporter inhibitors that has different function, but one of the important function that it blocks glucose reabsorption from the kidneys, and sort of glucose gets excreted through the urine. There are multiple trials looking at cardiovascular disease and renal outcomes and improvement in type 2 diabetes individuals. And that's the individuals who is this drug FDA approved for. It's now approved for some non-diabetes with improving outcomes, congestive heart failure and kidney outcomes, but nothing had been done in the outcomes for type 1 diabetes. And because of the risk of ketoacidosis, the drug is not approved in type 1 diabetes currently. So our trial focus on efficacy and safety. This is a multi-center trial funded by Breakthrough Type 1 Diabetes, or former Juvenile Diabetes Research Foundation. It's called Sugar and Salt Trial. Uses this medication, sotagliflozin. We are enrolling 150 individuals with type 1 diabetes with moderate to severe diabetes kidney disease with the protein in the urine over 200 milligram per gram and glomerular filtration rate reduced. And this is sort of our 3-year timeline for the study. And the last but not the least, I wanted to just, I mentioned at the breakout session about diabetes camps. Whoever is interested in diabetes, I would very strongly recommend you looking into this. This would boost your CV. This would boost your applications. It's an amazing experience. So this is through Diabetes Youth Families Foundation. I put here the website. There are camps in Southern California. There are camps pretty much throughout the United States. This is one of our picture of the camp from this past year, from 2024. Those campfires, and you can see our campers and the staff and the counselors. They are doing those skits all fun. They are making fun of us as medical staff and student and campers. Lots and lots of fun. And at the end of the camp, this is, I think, the last day camp. It's just all the tears because no one wants to leave the camp after this week of the camp. They so much loving it, and they made so many great friends. So I will stop there and feel free to ask me the questions.
Great. Well, thank you very much, Dr. Basina. I think we all enjoyed your presentation. Just a reminder, if anybody has questions for Dr. Basina, please ask them in the Zoom chat, and she can answer them from there. I'm Henry Barnes. I'm the Director of Education at SCD. Our next speaker received his doctorate in pediatric medicine with honors from the California School of Pediatric Medicine at Samuel Merritt University, then served his residency in pediatric medicine and surgery with the Department of Veterans Affairs in multiple hospitals across Los Angeles. His interest in preventing amputations led him to get his master's in public health from the Giesel School of Medicine at Dartmouth. He completed the Lawrence B. Harkless Fellowship in Limb Preservation at Keck Medicine of USC. He is currently a podiatrist at Keck Medicine of USC and specializes in chronic wounds of the foot and ankle, lower extremity biomechanics, and foot and ankle reconstructive surgery. His research interests include population studies in non-traumatic amputation prevention and healthcare disparities, along with biomechanics and foot and ankle reconstruction as part of wound management. He's widely published in peer-reviewed medical journals on topics such as diabetic foot epidemiology, diabetic foot ulcer and infection diagnostic imaging, and diabetic foot prevention strategy and policy. He is active in the American and California Pediatric Medical Associations and the American Association of Colleges of Pediatric Medicine, where he serves as a mentor. He also received the Arthur E. Heflin Award for Outstanding Research and the John and Janet Carson Public Health Advocacy Award, both from the American Public Health Association. Today, he will be discussing diabetic foot ulcer management from the lens of pediatric surgery. So without further ado, it is an honor to welcome Dr. Jiading Shih.
Thank you so much for the very extensive and wonderful introduction. Thank you so much. Share my screen. I'm in a hospital, so if there's happen to be like overhead announcement, I apologize. Here's my presentation. All right, can you all see my slides? Thank you. All right, so again, my name is JD Shih, currently assistant professor of clinical surgery under Division of Vascular Surgery and endovascular therapy at USC. So today my topic is diabetic foot ulcer EFU management from the lens of pediatric surgery. It's been a very informative morning, learning from immune system all the way to liver to population health. Mine is more going to be one-on-one patient and individual care. I have nothing to disclose for this presentation. So Dr. Armstrong, many of you may already know, he is my mentor and also my partner. We work together for the USC limb salvage program. So why is this topic important? This is why he found out. Every 1.2 seconds, somebody's having a diabetic foot ulcer. And every 20 seconds, someone with diabetes, unfortunately, will undergo amputation. And the incidence of diabetic foot ulcer is estimated to be 15 to 25% among patients with diabetes. And I think that number is only going up because recently CDC has published a publication saying that not only more people, but also the age group is decreasing. So why is this an issue? Because there's been studies showing once patients have amputation, it correlates to the five-year mortality rate, which is around 50%. It's a bit more complicated than that, not just the amputation that's causing I think that, let me rephrase this, amputation is not the cause of the mortality rate. It was the indication that a person's health is declining that leads to the amputation. Something else is already happening inside the body that leads to this very high 5-year mortality rate, even higher than some cancers. So, and that's my population. So the goal why we're trying to do limb salvage, one is if we can preserve their function, hopefully that preserve their cardiovascular capacity, so then they can continue to move, and so then they won't become more sedentary and they start developing pressure ulcers or other ulcers that leads to infection and unfortunately amputation or even death. So just quick review on the pathophysio of DFU, the foot ulcer. Dr. Paul Brand, who's the person really started to recognize this phenomena about diabetic foot ulcer. He thought it was a combination of this insensitivity, repetitive pressures, and skin breakdown that lead to this foot ulcer at the bottom of the foot. The other thing that he noticed is usually there will be an increased temperature before you see the skin to break down. And that's more explaining this inflammatory reaction first to this wound to develop, and then we see the wound to happen. So there's a quote from him: "The gift of a pen that when lost result in late complication of diabetic." So he really pretty much was the guru and the I don't want to say the founder, but like the person who really started looking into why this diabetic foot ulcer phenomenon is leading to so many issues later on. This is a review article from Dr. Armstrong in New England Journal of Medicine, and it really summarized why we see these foot wounds on the foot. So not only the loss of sensory that you see among diabetic patients, especially if their diabetes is not controlled, the loss of this protective sensation that leads to the insensitivity. Then because the nerve function is compromised, the intrinsic muscle inside of the foot would also change, and that leads to the hematose deformity, and that would result in the buckling of the buckling of the toe, increase the pressure at the ball of the foot, and that's why you often to see an ulcer at this area at the ball of the foot. And then at the same time, the autonomic system is also compromised, so the skin tends to be very dry, and when the skin's dry, it's very easy to break down. So once they start walking, they don't feel it, the callus builds up, then you start to have this wound developing underneath the skin. Eventually will break down and becomes a foot ulcer. And a lot of times they don't even know until the trail of blood at home, and then the family member is like, "Hey, why is blood on the floor?" That's a lot of times it's already happening. Sometimes they don't even know until there's some smell going on. So from my perspective, how do I manage this diabetic ulcer? If it's already happening, it's already there, how do we reverse this wound? So like the picture summarized earlier, if there's pressure at the ball of the foot, then my goal is to find a way to decrease that pressure. So then hopefully once there's no pressure, the wound will just heal on their own. So when I'm in the clinic, I look at the patient walking. A lot of times when they're walking from the waiting room to check the vitals, I already see their gait. That's already part of my physical assessment. And then once they're sitting and I do my non-weight-bearing exam, I can see if there's to correlate where that wound is. So there's a lot of biomechanic evaluation involved in diabetic foot ulcers. It's not a cut, just like on the leg or an arm. It's a very different ways to manage them. So this is a pressure map. When people walk, you see how certain area will light up to be more red. That's where you have the heel strike, and then you move forward, you push it off on the big toe, and that's why at the ball of the foot, a lot of time at the end, that's where the increase in the pressure is. And we learned just a few presentations earlier, the obesity is very prevalent among patients with diabetes. So just imagine the amount of pressure that each step that they take and don't feel that amount of pressure in that particular area under the foot can be very, very high. And so this is just another map on the right that to show you people who had amputation in the past, they may have different distribution of the pressure under the foot. So the following slides, I'll have some graphic pictures related to wounds and then some surgeries. So just want to give you a little warning. And this is just a picture to show you how when we push it off on the toe off at the end of your gait, how it's so we can move forward, and that causes the pressure ground reactive force plus the shearing force under the skin, and that often leads to something like this. This patient's one of my patients, another provider referred to me because they try everything else, just never healed. And then unfortunately at one point it was also got infected with treated antibiotics, but and he was only early 50s, and it's been battling this for like a couple of years. So we'll go back to this case a little bit, but that's just to show you how much pressure is in that particular area when the person is walking. So conservatively, we could take the pressure off by putting patients in a cast. There's something called a total contact cast. Unfortunately, something that you cannot remove. Obviously, if it's on the right foot, it cannot drive. So it's very burdensome, but it's very effective. It's supposed to be the gold standard to treat diabetic foot ulcer. When things are not possible, immobilized through total contact cast, then there's these removable devices to take the pressure off. So the patients can take it off when they're driving, put it back on. Ideally, they should treat this like a cast. So these are different options. If they still cannot tolerate that, then there's these post-op shoes. Obviously, you can see they're not as they won't take the motion off the ankle area as much, but you could take some pressure off from the bottom of the foot. If the forefoot pressure is too much, you can have this post-op shoe that has the forefoot offloading, or vice versa. You have the heel offloading post-op shoe. Just be careful with them because I have patients wear these and then fall. And many of them are on blood thinners, and that could lead to other complications. The other option is this felt pad, which is one of my favorites. Just put the felt on the foot and then cut a hole around the wound. So even the patient's walking, there's no pressure in that area. And it's very effective to take the pressure off. I use this a lot for younger patients because they have to work. So they can be in the shoe, wear this, at the same time still take the pressure off. It's very tough to tell patients, yeah, this one is not going to heal for 2-3 months and you don't go to work. That's just not possible. Other things like shoe modifications, it can be effective too. If the underlying cause, which the biomechanic part is not addressed, in my opinion, the recurrence is high. But in general, recurrence is very high because patients with the underlying reason is the neuropathy that cannot feel. That's not something that to my knowledge, we don't have the ability to reverse it. So unless we can bring back this their sensory function so that they feel there's a pain, they'll stop walking on it. Otherwise, the wound will tend to come back at the same spot or a different spot. This is a study that Dr. Armstrong published in New England Journal of Medicine and showing the recurrence rate sort of plateau in about 5 years, but still it's up more than 50% of chance that the patient will have diabetic foot ulcer again. So on the toe, like I mentioned, the hammer toe deformity, so the patient will have this wound at the tip of the toe. One way to address the pressure, something that we can do in a clinical setting, is just release that tendon at the bottom of the toe, the flexor tendon, and really just bring a needle, an 18-gauge needle in a clinic, prep the toe, and then just release that tendon. That will bring the toe straight. You can see a little bit difference from the position of the toe. The picture B show that toe is much straight and the wound is already healed. It's very effective. So this is how you should show you how to do the procedure, and literally that's the size of the incision.
Dr. Cody, get it? 3436. Dr. Cody.
The other way to prevent diabetic foot ulcer is the appropriate shoes. This is a very old picture. Nowadays, the diabetic these quote-unquote diabetic shoes are actually not looking all like this. A lot of them look just like New Balance, certain brands. They actually have specific lines for diabetic ulcer, patients with diabetes. This is a typical script that we'll write so the patient can get these shoes to fit it. And this is just how when we educate about patients about shoes, that they have to have certain width, depth, so then that the toes will be accommodated throughout the gait. There'll be no irritation. Remember, they won't feel a thing on the foot, and if there's any irritation, a lot of times they don't really know until they take the shoes off and socks off. Obviously, if you pick the style that they won't wear, then that defeats the whole purpose, and that just increases our healthcare costs. So when we try all the above conservative treatments that's not working, then I start to have a conversation with my patients, some surgical interventions. So this is a big toe ulcer that's been ongoing for a long time. And you can see how the toe has a hematoma on the big toe, and you can see how the lesser toes are also, you don't see the whole toe, you just see the tip of the toe. So what we did is we go in and I remove something called arthroplasty. So I remove the joint of that lesser toe. The big toe, and that will allow the motion back from the toe. And then in weeks, you see the picture underneath, that ulcer has been healed. And that was when I was in private practice in Northern California. And then when I left, she still has no wounds at that time. And this is another wound that can happen on the side of the heel. And you can see how this person's foot is tilting inward. And that's essentially how he's walking, because one of the tendon is weakened. So what we do is we transfer the tendon from the intertibialis tendon that inserts to the medial side of the foot, inside of the foot, to the outside of the foot. Now at rest, you can see how the foot compared to the top left versus bottom right. Now the foot even at rest is plantigrade, it's rectus. So when he's walking, he's not putting pressure on the side of the foot, and the wound will just heal. So back to this case that we talked about earlier, this non-healing wound, you can see how exaggerated this ball of the foot is under the big toe area. And so we did a surgery. We removed the two little bone under that area, and then I do a similar arthroplasty to the big toe joint. And this is, you still see the steri strip on the side, but the wound starts to close. And the suture's out, the wound is already closed, and he's not even, last time I saw him is about months ago, and the wound has been healed. He's doing his work, and I don't even know what he's doing now. This is another sorry about that. So this is another area that's very common to have a foot ulcer under the second toe. When there's increased pressure from the buckling of the second toe, you can see in the middle picture, the second toe kind of curve up a little bit. And because of that deformity, that would put pressure back at the ball of the foot when the patient's walking. And this is something called floating osteotomy. We make a tiny little incision, make a cut at the surgical head of the metatarsal bone, and just let it fly. And this is, I think it was a month out, it's already gone, and just some callus, we can shave it down and put him in the appropriate shoes. This is a little bit more extensive. This person has something called a Charcot foot. It's very common. Well, let me rephrase. It's not quite common. It's estimated about 1 to 2% of patients with neuropathy that can have Charcot foot. And so he literally walk on the side of the ankle. And this, if you put a probe, that will go directly into the joint. So it's a very complex reconstruction for this case. We put an X-fix on, we have to manage the infection first, and eventually we fuse the joint, and then he's in a brace, and then walking a brace. But the wound on the side of the ankle, it's not there anymore once that deformity is corrected. So in summary, neuropathic ulcers, it's especially diabetic ulcers, it's a result of peripheral neuropathy and increased pressure in that certain area and the shear force. So to address that, offloading is very important. To find a way to kind of think about how to take that pressure off. Shoes, accommodating inserts, it's the primary way to prevent these wounds to come back. But there are times, unfortunately, reconstruction and surgery is necessary to prevent these foot ulcers to come back. So that is my team, US Vascular Surgery. It's my email underneath. Thank you for listening. Feel free to reach out to me if you have any questions about dietary or things we do. Thank you much.
Thank you very much, Dr. Shih, for your amazing presentation. If you have any questions for Dr. Shih, please ask them in the Zoom chat and he will answer them from there. I'm Sam Akumbo, the co-director of local involvement at SCD, and our next set of speakers will be discussing diabetes technology, more specifically, diabetes technology research encompassing the scientific investigation and development of devices, software, and systems designed to improve the management and treatment of diseases, of diabetes. Our first speaker obtained her MS and PhD in electrical and computer engineering from Georgia Institute of Technology, and her BSc also in electrical and computer engineering from the University of Texas at Austin. Before joining Dartmouth, she was a Rice Academy postdoctoral fellow at the Scalable Health Labs. She is currently an assistant professor in the Department of Computer Science at Dartmouth College. She founded and directs the Augmented Health Lab at Dartmouth College, a consortium of interdisciplinary researchers working together to tackle grand challenges in healthcare. Currently, her research focuses on digital and computational health, wearable technology, and data science, with a focus on developing human-centered computing solutions that enable personalized health. Today, she'll be discussing making sense of diabetes with technology and data-driven analysis. Without further ado, it is an honor to welcome Dr. Temporal Prioleau. Sorry if I mispronounced that.
Not a problem at all. Thank you so much for the warm introduction. It's been amazing to be a part of this. So I'll start by sharing my screen. I assume you can see my screen? Yeah, sounds good. Alrighty, so I'll go ahead and get started just to be cognizant of time and make sure to leave time for questions, whether it be in person or via chat. So based on the introduction, my name is Prioleau. I am an assistant professor in computer science at Dartmouth, and today I'll be talking to you about making sense of diabetes with technology and data-driven analysis. So here is some background, but we've already learned a lot of this, but diabetes is a prevalent chronic condition both in the US and globally. Very simply, from my end, diabetes is characterized by impaired glucose metabolism. And what that may look like is for someone with diabetes, they are prone to having high blood glucose, aka the blood glucose going extremely high, hyperglycemia, meaning above the target range, or low blood glucose, so going below the target range. There are many reasons for this, whether it be type 1 diabetes or type 2 diabetes. But at the core, that would be one description of what diabetes is. The body is not able to control blood glucose within that healthy and target range. So to navigate kind of the complexity, there's so many things for someone that already has diabetes. There's so many factors in life that affect management or that affects what someone's diabetes might look like, whether it be On the one hand, there's glucose, but then there's also nutrition and lifestyle factors, physical activity, stress, even sleep behaviors, age, geographical location, socioeconomic. There's so many factors that combine and play a role in what management might look like. And there's something called digital phenotyping. And what that simply means is trying to measure some or as many of these things as we can so that we can take data that comes from these different factors, put it together for data analytics, integrating that, combine that with AI and machine learning to be able to support or get towards what is called personalized health or personalized interventions. So that's some of where my work is, and that's some of what I will speak about today. So when it comes to diabetes, I guess I can start by talking about digital health technology. So in my opinion, diabetes is ahead of the curve when it comes to technology for ubiquitous and continuous monitoring. So if you're familiar with some of the technology, the standard is your glucometer. So just like the finger prick, you can grab one at CVS or in Walmart, whatever local stores. And then we've already heard about continuous glucose monitors. These are somewhat wearable, minimally invasive wearable devices that continuously monitor blood glucose, usually every 5 minutes, insulin pumps, and then your standard activity trackers. So these are all the devices, technology that monitor some aspects that is relevant to diabetes. So in the US, kind of using the US as just as an example, the status quo is, let's say 40% of people with type 1 diabetes use CGMs, and around 65% of people with type 1 diabetes use insulin pumps. And if you notice, I'm talking about type 1 diabetes here, although it does affect type 2 diabetes and others. But the reason I give these numbers is just to give an estimate of the number of people that use CGMs, for example. So this equates to around 600,000 people in the United States using a continuous glucose monitor. And what that means is data is being collected approximately every 5 minutes from 600,000 people in the United States. What that means is there's a lot of data that's being collected, and hopefully there's a lot of data that we can leverage to learn more about management, to learn more about obstacles or challenges, or what people's diabetes measure might look like as they go about their daily lives. So this leads to kind of where some of my work comes in, is how might we use everyday technology to understand and inform diabetes management? For my lab, I guess I could think of it, you could think of it as opportunistic use of digital health data to understand and improve diabetes outcomes. And they're kind of three aspects that I'll touch on. I'm going to touch on very briefly given that it's a short presentation. So one thing that we work on is what I call context-aware sensing for digital health interventions, and I'll give an example of that. Another aspect that we work on is wearable data analytics and AI for personalized care. And then the last one, which is a little bit more recent, is focused on reproducibility in machine learning research for health, but particularly for diabetes. So I'll give examples of each of these and before I conclude. So one of the projects that my lab has been working on over the last at least 5 years is this project called HealthMine. And at the very core, the goal of what we're trying to do is integrate data from your continuous glucose monitors, which is useful for understanding what the blood glucose looks like as people go about their daily lives, as well as activity trackers, and then what can be called ecological momentary assessment, or just user logs, basically things that they are not particularly sensors to objectively monitor certain factors, for example, stress or diet. They are not particular sensors that can be used for monitoring that. So giving a user or a person the opportunity to log various things that are relevant to their diabetes management. So this is a mobile app that has been developed in my lab, and the goal, kind of what we're leveraging it for, is to enable longitudinal studies in the diabetes space to uncover relationships between individualized lifestyle factors and outcomes. And then lastly, most importantly, let's say, to support digital interventions that can support improvement of outcomes. So one project, and this is one project that started last year, it's called the Reflect T2D, and these are some of the collaborators that we work with. And this project basically leverages HealthMine, which is a software solution that has been developed in my lab for deploying a micro-randomized intervention study amongst a population of people with youth onset type 2 diabetes. And very simply, the goal is to try to help people leverage data from digital technology, so the CGMs. So when people are recruited, they're given a CGM, they're given a Fitbit, and they're given the mobile app, and they're asked to use it. But every so often through the length of the study, the 3-month length of the study, they get feedback based on their own data, if their blood glucose was high or low, and how that might relate to their physical activity, or how that might relate to their nutrition, aka what they ate at different points. Given feedback to help make connections between what they're doing on a daily basis and what their blood glucose looks like. The goal is to increase knowledge of how the decisions that we all make on a daily basis, how that influences blood glucose, and what that, to make those connections so that we can hopefully make changes. And then towards the second part, which is relating to wearable data analytics, here are just some examples of different papers that have been published by my lab. I'll just give some examples of some of the things that we've done. So one paper focused on trying to understand seasonal variations of glycemic trends using data from CGMs. So basically, if we look across the full year, the length of the year, as lots of people using CGMs, we basically took retrospective data, observational data from lots of people that have used CGMs. And the questions that we try to ask is, when do people have better management? When do people have not so good management? Just looking across the year. And some of the things we learned, and maybe this might be obvious, but it's less studied, is that, for example, during the winter months, people tend to have not so good management compared to whatever their own average is of managing diabetes compared to during the summer months. And the goal of this is, if we can learn this from data, then we can hopefully understand times for intervention, time to provide support. So that's an example. Here's another example of something else that we've done, leveraging data from wearable diabetes technology. So we recruited people that have been using insulin pumps for lots of years. And then we basically took the insulin pump data. And one of the things that's very key with insulin pump and use is people need to log. And even if this is also relevant even for hybrid closed loop systems, you still need to log food related meal logs, I guess, or carb inputs. So what we try to do is we try to say, well, if someone has been logging their meals for months, maybe even years, can we take that data and try to learn when people eat on a personalized level, such that we can provide support or nudge people to remember, because maintaining blood glucose post meals, maintaining normal blood glucose post meals is definitely a challenge. So we took historical data and tried to leverage that to learn patterns of when people eat, to be able to give nudge them or reminders as to when for taking or getting bolus insulin, which is the insulin that would be related to meals. So these are just some examples of things that we do and we have done leveraging data. But one of the things that was mentioned that, you know, one of the organizers wants me to talk about is this work related diet trend. So one of the things, when I started in the diabetes space, one of the things from an engineering and computer science perspective, that's a challenge, is getting access to data to study. So one of the things that we've done within my lab is we've basically created a data set. It's called Diet Trend, for more of the engineering, computer science folks, the people that develop the algorithms to leverage this data and other data sets to build algorithms that can hopefully be helpful to support diabetes technology and diabetes management. So specifically, I'll tell you about the Diatrend data set, but I'll also make sure to tell you about other data sets that are similar. So Diatrend is an open source yet controlled data set. When I say controlled, meaning each person that is interested in this data set needs to kind of go in and put their name, create an account, and request the data set. So it is controlled, but it is open source. The Diatrend data set includes data from 54 people with type 1 diabetes, and it includes, you know, more than 27,000 days of CGM data, more than 8,000 days of insulin pump data. And the goal of releasing this data set is to support the data scientists with developing algorithms that can be relevant for the diabetes community. So far, it's been released. The diatrend has been provided to over 50 researchers, and more people are requesting it. So if anyone is interested, feel free to just, either scan the QR code or just search diatrend, and you should be able to find the data set and request it if you're interested. And then to give an example of some of the things that we are doing as it relates to reproducibility, from if you call it machine learning perspective, one of the most common tasks in the diabetes space is blood glucose prediction. So lots of people and labs work on blood glucose prediction. And the reason that's important, there are many reasons why it's important, but if we think about someone with type 1 diabetes, blood glucose prediction is important because it's the very core of automated insulin delivery systems. Basically, the algorithms predict blood glucose, and that helps inform how much insulin to give at a certain point. But one of the things that we've been doing is says, well, let's take a look at a lot of the models, deep learning models that have been developed in the last 7 years for blood glucose prediction. And then let's re-implement those models, test it on the original data set that it was built on, as well, which is a lot of models and methods have used what's called the Ohio T1D data set, but then also tested on other open source data sets. So Diatrend is one, and then another one is T1Dexy. And let's see how reproducible these methods are. And one of the things that we found is that there is often, in many cases, a lot of difference between what is published as the performance of methods compared to when it's reproduced, the reproduced results. So we're bringing this to light with the goal with the goal of trying to reduce or increase reproducibility of methods, point out other data sets, point out gaps and reasons why methods might have issues with being reproducible. Another thing that we're finding, I would say this with caution because this is still early work and it's not published yet, is that we've actually found across lots of methods that have been published, there tends to be, or what looks like what we're seeing, is a difference in prediction accuracy across genders. We don't understand the full reason why that is, but we're seeing that methods across males have lower error compared to, when it comes to blood glucose prediction compared to females. So some of the things that we're working on now is trying to understand some of the reasons why a lot of these deep learning methods have, if you call it gender bias or differential performance across genders. So the last thing I want to point out is something else that we're doing is we're also just collating lots of, in the last few,
Yeah, that's a lot. I think that's like the hard.
in the last few years, there have been several other open data sets that have been released in the diabetes space. And this is something we're working on now. So I put the data sets here. That way you can take a screenshot if you're interested in data sets in the diabetes space. So these are recent data sets that are large and that are well tested, recorded from real people, not simulated data. Data sets that have been collected from the US, from China, from Spain, from Chile, data sets that include type 1 diabetes, pre-diabetes, type 2 diabetes, and no diabetes, as well as the sample sizes. So some of what we're doing is just collating these data sets together and even doing a comparative analysis across data sets to try to bring to light what each data set might be best useful for when it comes to developing models that can be relevant for the diabetes space. So with that, just my summary slide. From my end, there is immense opportunity for leveraging user-generated data to transform our understanding of chronic conditions, including diabetes. A lot of people already use CGMs, already use insulin pumps. So there's a lot of data out there that can be useful for better understanding management and pinpointing challenges that people might be having. Overall, I would argue that digital health data is significantly underutilized, and there's a lot of room and a lot more work to be done. So with that, I have lots of faces and lots of people to thank. So I say thank you for listening. And if you have any questions, feel free to reach out to me. My email is here, and this is my lab website.
Thank you for those amazing insights. If you have any questions for Dr. Prioleau, please ask them in the Zoom chat and she will answer them from there. Our next speaker and last speaker for the day, Dr. Francis Doyle, provost at Brown University, unfortunately had to cancel last morning as his university is subject to a $510 funding cut as of a few days ago. Now, as the U.S. Diabetes Conference has come to a close, we would like to have a short fundraising segment to raise funds for our student organization at USC. This will go to support our organization's activities. So here's the QR code that you can scan, which goes to our Venmo. And if you would like to donate or don't have a Venmo, you can email us at diab@usc.edu for other ways to donate. We'll give you guys a few minutes to scan this QR code. And thank you for your donations if you choose to do so.
All right. Thank you everyone for coming to US Diab Con. We hope everyone had a great time listening to diabetes experts from across the US and networking with them as well. I'd like to thank all of our exec members, Aryan, Avi, Ashwin, Tiara, Henry, Sam, Adolfo, and our general member breakout room moderators, Jeff, Mohammed, Suresh, and Michael, and all of our other general members. I'd also like to thank our advisors, Dr. Alyssa Dominguez, Dr. David Warburton, and Dr. Tyler Mason. Lastly, I'd also like to thank all of our amazing speakers today. All in all, we hope you were able to learn a lot and connect from one another. One day, diabetes will no longer exist, and loved ones, communities, etc., will no longer have to suffer from it. It is with this sharing of knowledge and connection within the diabetes community that that day will come sooner. With that being said, US DiabCon is now adjourned. Thank you so much.
Yeah.
Thank you.
Thank you so much.
Thank you.
Thank you.